Welcome back. We have some coming fantastic talks here today. So I would like, oh, by the way, I'm Susu. Gotcha. Introduce myself. I'm basically in USA now, and I'm part of the members from SSE GSEC. And I would like to welcome the next speaker, Ruchishree Konhar. Ruchishree is a PhD candidate at the Institute of Genomics and Integrative Biology, New Delhi. She studies the evolutionary patterns of nuclear and organelle genomes of diverse organisms. Today, she will share with us some insights into the fairy tale life of an Indian pincher plant and other carnivorous plants. So it's your time. Ruchishree.
So hi everyone. So the title of my talk is The Devil's Basket: Insights into the Fairy Tale Life of an Indian Pitcher Plant and Other Carnivorous Plants. So when we talk about plants, what is it that comes to our mind? How do I get rid of this? So when we talk about plants, what is the thing that comes to our mind? Plants are basically green organisms and they come in a variety of shapes and sizes. And when we talk about our dependence on plants, that is human beings, we are heavily dependent on plants because they serve as our source of food, our source of clothing, our house and medicines and a lot of things. And also when we talk about the animal kingdom, the plants basically feature at the base of the food pyramid. That is, they have always been the producers. And but when we talk about carnivorous plants, that is where the tables turn. So what happens in case of carnivorous? What happens in case of carnivorous plants? The plants, instead of being the producers, start becoming the consumers. So the plants trap small insects and tiny rodents and digest them and eat them up. So carnivorous plants basically grow in nutrient poor soil, and for a plant to be called carnivorous, it should have three basic properties. First, it should have the adaptations necessary to lure and trap prey. Second, it should have the ability to digest the prey. And third, it should have the ability to absorb the nutrients from the digested prey and use it for its own growth and reproduction. And generally what is seen is plants use modified leaves for doing all these activities. So when we talk about angiosperms or the flowering plants, there are about 300,000 species that are reported, and of these only 600 have evolved the carnivory trait. So there are different trapping mechanisms the plants have evolved. So the first one we'll talk about is bladderworts. They use these tiny suction bladders to suck in their prey. Then we have something called butterworts. These use this mucilaginous substances on their leaves for trapping the prey. And sundews, they use these mobile tentacles to fix their prey onto a place and then digest them. Then we have the snap traps. This is something I think we all are pretty familiar with. And finally, we have something called the pitfall traps. When we talk about pitfall traps, they are again spread in 5 different genera. Cephalotus, that is the Australian pitcher plant. Then we have the Darlingtonia, Sarracenia, and Helianthera. These are your American pitcher plants. And then we have Nepenthes, that is the Asian or tropical pitcher plant. And when we see the spread of these carnivorous plants, that is the different trapping mechanisms they use to derive nutrients from their prey, they are spread across the angiosperm family. So this is a classic case of convergent evolution. So when the plants were faced with the challenge of growing in a nutrient poor soil, the solution they came up with was trapping and deriving nutrients from their digested prey. So now when we come to Northeast India is basically comprised of these 7 states, and this is one of the most floristically diverse place in Asia, and it has a unique topographical profile. We have the Brahmaputra River Valley over here to the Kanchenjunga Mountains over here. So this gives rise to a very unique region for the biodiversity that we see here. And when we talk about carnivorous plants in this region, in India there are a total of 44 species of carnivorous plants that are reported, of which there are 21 that can be found in the Northeast India. And when we talk about the state Meghalaya over here, we have two species of Drosera, one species of Nepenthes, that is the Nepenthes khasiana, and we have three species of Utricularia that grows here in Meghalaya. So what we did was we isolated the marker regions ITS and MATCAVE, nuclear and chloroplast marker regions from the species that grow in Meghalaya. And this is a picture of Nepenthes khasiana in its natural habitat. These are the developing pictures. That is, when it develops, initially it is closed like this. When it grows into a adult size, so before it opens, it fills up its belly with the digestive juices, and once it's ready, it opens its lid for trapping the prey. So we took the marker regions, and the first thing that we did was we did a bicy and phylogeny, and based on the from this we can see that we saw a very good grouping based of the OTUs into different families like the Droseraceae, Nepenthes, Ancestocladidae, and Dionchophyllaceae. So with this, we went ahead and did a time tree estimation. So for the time tree estimation based on fossil and literature, we used the time constraints for this node over here. For this node over here to be somewhere around 55 to 38 million years ago, and the most recent common ancestor for all the Drosera species to be somewhere around 5 to 22 million years ago. And we also did a ancestral area reconstruction for the species. So if you look at the spread of Nepenthes, these are all found in Asia. And if we go back a little, and if we see the base of this Nepenthes species, we see that it is found in Asia and Africa. So if we combine our results from the ancestral area reconstruction and time tree, we can probably explain the evolution of the genus Nepenthes and the current day distribution that we see today. So Nepenthes is probably originated at a time when the Indian plate, the Australian plate, and the Madagascar plate were all clubbed together over here. And with time, after the Nepenthes species originated, sometime around the Australian plate moved over here, the Madagascar plate moved over here with its Nepenthes species, and the Indian and Sri Lankan plate with their own Nepenthes species traveled all the way over here and collided with the Eurasian plate. So before the arrival of the Indian plate over here, Asia did not have any, this region did not have any Nepenthes species. So with the arrival of Nepenthes over here, and here, this led to the radiation of the Nepenthes species, and this is the diversity of the Nepenthes genus that we see today. So this makes it a very important species in the evolution of the Nepenthes genus. So if we talk about Nepenthes khasiana, this is the only pitcher plant that is found in India, and it grows at an altitude of about 100 to 1500 meters, and this has a very, a very localized distribution in the hills of Meghalaya. And it generally grows in rocky mountains. In rocky places, it has a stunted growth, and in forests, it acts as a climber. That is, it grows over trees and bushes. And if we talk about the other importance of Nepenthes khasiana, it has been traditionally used as a herbal drug by the locals for treating various diseases like diabetes, urinary disorders, cataracts, skin infections, GI diseases, and other gynecological problems. And apart from that, it produces a protein, digestive protein called nepenthesin. When nepenthesin is used in conjunction with pepsine, it shows a drastic reduction in intestinal inflammation. So this combination is something that is being explored for the treatment of celiac disease. Apart from that, the hypoglycemic and hypolipidemic effects of the pitcher fluid has also been studied, and it also produces a compound called plumbagin, which has been traditionally already been known to be anti-cancer, anti-malaria, and insect antifidant. And apart from that, it produces, upon induction with chitin, it produces droseron and 5-methyldroseron. These have potential antifungal activities against many of the major plant and animal fungal pathogens. And when we talk about its importance from the biodiversity and evolutionary point of view, like we have already discussed, is probably a paleo-endemic species, has a unique habitat, and it has been classified as endangered and threatened by the IUCN. So what do we know so far about Nepenthes khasiana? So far, all the studies till 2019 focused on isolating single genes or single proteins, and in 2019 came up the first transcriptome of the species. And since there was no genome of the species, we thought we'll look at the genome as well. So for this, we did the genome assembly and annotation of Nepenthes khasiana. And from here, I could annotate about 28,000 enzymes in the Nepenthes Khasiana genome. And now with the advent of more and more sequencing technologies and the lack of fossil data, if to understand the evolutionary history of any organism, we are looking more towards the genomes than we are waiting for the fossils. So if we talk about the genomes, then as of today, there are 8 carnivorous plant genomes that are available, apart from the Nepenthes khasiana that we have sequenced and annotated. If we look at the genome of Genitia and Utricularia, we know that these are the smallest genomes in the angiosperm family. And even though they might have gotten rid of the many of its genome, but the gene content of these species have not gone down. They are pretty much at par with the other angiosperm species. So just to check the digestive, this is a list of the digestive enzymes that have been reported so far from all these species, spanning across different families. So I checked for the presence of these enzymes in my genome, that is the Nepenthes khasiana genome, and indeed all of them were present in my genome. So this indeed confirms our theory of a convergent evolution, that all the plants that have evolved carnivory, they have basically used or repurposed the same genes for digesting their prey. And now when we compare the Nepenthes khasiana genome with another pitcher plant, that is a cephalotus genome, and other non-carnivorous plants, many of the genes are shared by all of them. That is, the basic functionality remains same, but it is a few genes that have been repurposed, or there have been a gene expansion and contraction in a few families that have led to the evolution of carnivory in few plants. Now, apart from the nuclear genome, we also pulled out the mitochondrial genome. For the mitochondrial genome, the plants have the largest genomes in the eukaryotic organisms, in case of eukaryotic organisms. And even though the size has expanded, the number of genes pretty much remains same. So for this, I used the MATK gene as a seed for my assembly. And I have assembled and annotated the Nepenthes khasiana mitochondrial genome. So this is about 900 MB in my case. And for the chloroplast genome, we use the RBCL as the seed sequence for the assembly and annotation. And I ended up with a 156 kb genome of Nepenthes khasiana chloroplast. Now, unlike the mitochondrial genome, where only 4 mitochondrial genomes of carnivorous plants are available, including mine, there is not much we'll gain from the comparative genomics of these species. But when it comes to the chloroplast genome, we have 35 chloroplast genomes available for the carnivorous plants, spanning across all the family from which carnivory has been reported. So if you look here, the first thing that we
Excuse me,
note.
3 minutes.
Yeah. Okay, sure. So the first thing that we see here is the grouping of the species based on the carnivorous plant genome. So the species tree is well recollected even when we use the whole CP genome. And another interesting thing that to note here is when we talk about these two families, that is Lentibulariaceae and Rosaceae, these have different trapping mechanisms that we see in the same family. So one study that is ongoing is we are trying to look at the pan genome based on at the family level, and by grouping the trapping mechanisms to see if there is any pattern that we'll see when we do this. And with this, I'll end my presentation. So when we think of plants, are all plants harmless? Well, they might not be, but if you are an insect, it could be. So this is basically the Nepenthes khasiana pitcher plant. Locally, the Garo people call this Nimang coxi, which basically translates to devil's basket. So if you are an insect, so you might as well be careful. And I would like to thank the organizers, SSC and ISCB, and my lab members. If there are any questions, I'll be happy to take.
Very interesting talk. Thank you.
Thank you.
Let's wait to see if we got some interesting questions from the audience too. I like the last slide. By the way, I'm an entomologist. I study bugs. It's very interesting that
bugs.
Yeah, it's very interesting to see that they all produce chitinase because they hunt for insects. Okay, we have a question. Oh, is that a UV image for the last slide?
Yes, that is. So this is Nepenthes khasiana, one of the trapping mechanisms of Nepenthes khasiana that is being showed here. What Nepenthes does is it uses UV blue light fluorescence in their rim to trap to lure the prey for trapping.
We have time for another question. Well, so thanks for the great talk again, Ruchishree. And if you have questions, welcome to always just type in the Q&A box or the chat box. And I believe our speakers will take their time to respond, to give you some responses. Thank you.